This is accelerating single-cell bioinformatics with n-dimensional arrays in the cloud, which is a lot. The three important ideas are that single-cell sequencing is a new technology that's very interesting from a scientific point of view, and it poses some computational challenges related to storing and processing array data. And this being 2018, we want to do that in a way that works on public clouds and is parallel where appropriate. So this work is being done at Uri Laserson's lab at Mount Sinai on a grant from the Chan Zuckerberg initiative that's part of the Human Cell Atlas. The Human Cell Atlas is like a multi-year, multi-institution effort to basically make sense of this single cell data that's going to be increasingly important and proliferated. You can kind of think of it like the Human Genome Project, but like 20 years later, and in response to a different kind of sequencing that's like being developed and generating interesting data that we want to standardize around the processing of. And also my colleague Tom White in particular did a lot of the work that I'm going to be presenting here. Okay, so agenda is basically, I will describe the sequencing breakthrough that's pretty interesting, talk about how we are storing the data. The existing tools are using HDF5, which is a container format that's been popular in a lot of scientific domains for like 20 years. Has some rough edges in the world of like distributed processing. There's a new technology called Zarr that we are mostly encouraging adoption of instead of HDF5, but also doing a lot of work to try to support both, because people have existing data in HDF5, and so we'll go into that stuff. We want to parallelize the analysis of this data, and there's a bunch of tools, that we've been working on to do that on various distributed backends that exist that are pretty interesting. And then I've worked on a n-dimensional array library in Scala for that implements the Zarr specification that we're trying to encourage people to use, and that compiles to JVM and to JavaScript, and will like help to just have a healthy multi-language ecosystem around this like format that we're evangelizing. So quick kind of silly exercise. We'll just go through some basic biology that's relevant to appreciate the breakthrough on the scientific side here. So the information that powers life goes through this process of being in DNA, which is like the cold storage, through RNA, which is like more volatile, memory-like, and ultimately to proteins, which like actually affect the world. And at every stage, it's kind of the same information, just encoded in different ways. Going from disk to memory, you have very identifiably the same information, just in a different physical substrate. You typically have more information on disk being stored than you're using at any given time. An interesting question is like if you're analyzing a biological system or a computer system, what kind of information about which stages of this process would you like to be able to read to answer different questions? And there's certain questions where you'd want to just being able to see a full snapshot of a disk image of a machine would be useful. In other cases, you'd want to be able to grab what's in memory and actually being executed, and then the actual pulling the CPU buffers and registers might be useful for some things, but a little bit too low level for other things. So to date, we've mostly only had the ability to read the disk and the memory of cells, and even that with some pretty severe limitations on what we're able to do. And so some of those limitations are being lifted, and that's kind of the breakthrough that's happening here. So another kind of like different way to make a similar analogy is that the DNA is like all of the apps that you have installed on your computer. You might not be running most of them at any given time, but they're there. And given a lot of machines that were even booted from the same disk image and have the same apps installed, they could run different programs and manifest in the world in very different ways. So you could, for the same pool of like applications installed, you might have a machine that's behaving like a web server or a graphic design workstation or a email and video chat client or a development workstation. And then the things that those running programs actually do to interact with the world are analogous to the proteins that actually comprise different types of cells that may all have the same DNA, but they're very different shapes and interact with the world in different ways. So that all being said, the essential breakthrough here that is ushering in this, is the animating idea for the human cell atlas, is that we are able to move from bulk sequencing to single cell sequencing. All of the genome sequencing that you've heard of to date over the last several decades has been bulk sequencing. We take a population of cells and we can sample from the population average of their DNA or their RNA. So you can imagine you're able to interact with a bunch of servers and get random snapshots of disk or memory, but you don't really know which server it came from. You have no ability to resolve at the single server level. It just makes things harder to identify if there are important subpopulations of the servers that are acting as web servers or graphic design workstations or what have you. And so through some clever chemistry tricks, we are now able to actually say, here are the exact populations of cells that exist, here are the programs that they are running, and even here's how they are oriented with respect to each other spatially, to indicate which types of cells are talking to one another, what types of cells even exist, etc. The clever chemistry trick is basically a pre-processing stage of the cells before putting them through the usual bulk sequencing pipeline. And this pre-processing step essentially puts every cell into a little oil droplet along with a barcoding bead that you kind of just run the cells and the beads through this in very dilute concentrations through this droplet mechanism. And most of the droplets end up empty, or they just have a cell but not a bead, or a bead but not a cell. But the important thing is you will get a maximum of one cell in a droplet and maximum of one bead in a droplet. And you take the droplets where you've had this successful isolating of a single cell with a single bead, and the bead kind of degrades the cell and attaches its barcodes to the cell's RNAs. So that then when you pool all the cells back together and you start sampling from that population, all of your samples have this per cell barcode attached to them, so that you can now actually say which RNA reads came from the same cell as which others, if that makes sense. And then essentially we cross the boundary from wet lab to dry lab and data analysis at this expression matrix, which is basically cells by genes. For every cell, how much of each gene did it express, which is how much of each program was it running. And then from there, we want to cluster the cells and do a variety of things to infer what profiles of types of applications we're running on these cells. I don't know if can I click? Yeah, cool. This is just a short video of kind of what that looks like. This is one solitary cell going through this pipeline, and the thing is just snapping off droplets, and what's missing here would be also the beads coming through and them getting snapped into a droplet together, but it's a basic idea. Cool. So the technology to be able to do this has been maturing over 5-ish years, and we've been able to push more cells through these pipelines, and so the Human Cell Atlas is a response to that. And you can see just kind of exponential increase in the number of cells. The largest data set, public data set now is about 1 million cells, and we're expecting many more over a period of years. And everyone's very excited about the ability to just understand these systems of cells better by knowing actually what the specific cell types are and inferring what they're doing and stuff. Okay, so then the data analysis. As I mentioned, the fundamental unit is basically the expression matrix. It's cells by genes, and the genes are basically fixed. If you're always looking at human data, there's like 30,000 genes, and then however many cells you want to push through these experiments. So I mentioned the largest one is like 1.3 million cells. It's like 40 billion entries, but it's mostly sparse, so it's maybe like a billion non-zero entries. Like most cells are not expressing most genes. And the two high-level classifications of kinds of workflows we want to run are like pre-processing and quality control, normalizing the entries in this matrix per cell or per gene. And then there's the more interesting downstream stuff is like different flavors of clustering to identify what types of cells exist. There's popular tools that exist and have very healthy open source ecosystems around them that are written and maintained by the labs that are doing a lot of these sequencing experiments, and they've made very high quality tools that like do all of the analysis questions that they want to answer about this data. The most popular ones are written in Python and R and use different HDF5 schemas as their like persistent storage and archival storage of this data. So as these data sets have grown, we want to be able to process them out of core more. And so most of the tools that I was talking about have some ability to do this. It's been bolted on a bit. They're more used to just paging the entire data set into memory and just doing NumPy things to it. And in particular, we want to be able to process these things in parallel and in clouds. And HDF5 has some issues doing that. So we've kind of backed into having to think about this problem of how to store and process multi-dimensional array data. So HDF5 is a very interesting project. It's been like standard in the scientific community for 20 years and widely used. It's basically a way to have like lots of data in a single file that has like a virtual file system inside of it, and there's like a directory structure within this file, and then the files within the file are data sets, which are just multi-dimensional arrays of a given type. And then it has various other like organically evolved features on top of that where things can link between each other, and there's like a lot of metadata of different shapes that can be attached to other things. And so there's a lot there, but that's the essence of it. It's kind of a side note is that it's not really a file format, it's like a meta format. Like you can, like in each scientific domain, they kind of decide what they're going to pass around. Like there's kind of like a schema you have to standardize on of like what directories with what names and what data sets with what names do we expect to find inside of one of these files, if it's geospatial data or single cell sequencing or something else. So it's more analogous to like choosing a database system, but then still wanting to standardize around like what tables and what schemas should exist in those tables. There are some difficulties that have become worse over the years, I think, with using HDF5. It has a very complicated spec, and there's basically one enormous reference implementation in C that has been wrapped in many other languages, but you're always essentially using this one implementation that's by like the main group, the HDF5 group, and it's not like what I or others would consider a super healthy open source project. Like it's not really that actively developed widely in the community, or like doesn't have a good like issue tracker kind of. I don't know, it just doesn't feel like the kind of ecosystem that I'm more used to and comfortable working with. And in particular, the C implementation like does not. It only reads from local files. So all these other languages, like Python has good libraries for interacting with Google Cloud Storage or S3, but you can't inject those anywhere because you're just calling this C library that's like hard coding like local file system I/O. We and others have tried to use fuse adapters to get around that. They never seem to really work, and it's kind of not really the way you'd want to do this anyway. So it's just kind of hard to deal with. And then the parallel processing story is also limited partly by just being stuck with a local file system. And also the idea of having these large data sets be just single files on an underlying file system poses problems for how you would like write one of these things with like 1,000-way parallelism. Like that's just like not really feasible in most file system-like environments. So there's kind of an emerging consensus in several scientific domains that are wanting to use something that's a little more natively parallel and cloud friendly than HDF5. So Zarr is the thing that we've been most excited about. It's a new, much simpler implementation of the same kind of idea. You have like groups and data sets that are roughly like directories and files that can be nested. And in Zarr, the files or data sets are themselves a directory on some underlying file system. But you could use just an arbitrary key value store instead of a file system here. But you can see in the lower right what a two-dimensional array looks like on disk in Zarr. There's like a Z-array metadata file that shows you the shape of the two-dimensional array and the shape of the chunks that it is partitioned into, and other information about the compression that's been used, which is pluggable. And so, and then you have like the files that are each chunk, 2 chunks on each axis, 4 chunks total, and those chunk files are just like 10 by 10 signed integers in this case. So it's just like 400 bytes run through zlib, and that's the file. It's fairly like straightforward how to interact with this, and you can easily parallel write these things because like, a worker can just write 1 chunk very easily, and likewise to read them. So it's a very simple spec. It removes a lot of cruft from HDF5. There's a reference implementation in Python, and then there's independent implementations in C++, and I've worked on one in Scala that compiles to the JVM and to JavaScript, like I mentioned. And it's a new and exciting community. There's not like baggage and legacy code that we have to worry about, so we can just kind of make it be what we want it to be. On the other hand, it's like missing essential tooling in some cases that we need to write, which will become our baggage and legacy code someday, but it will be ours. And well suited to our particular use case, I guess. It's being somewhat officially adopted by the Human Cell Atlas for storing and processing this data. So one other like notable entity in this space is the common data model and the implementation, the main implementation of it called NetCDF, which is an existing years old, pretty established standard that's like also a kind of simplified abstraction over HDF5 and other things like it that have this like nested groups and data sets that are multidimensional arrays kind of structure, some amount of extra features on top of that. It's like basically a subset of HDF5, although like kind of not technically, and so there's just some, but it is interesting because there is a Java implementation that works for all of these backends, including the parts of HDF5 that are supported by this common data model. So it's kind of like a second implementation of the HDF5 spec, but just some of it. So that'll be relevant later. Cool, so I'll just talk about what we've done so far and where we're going. So we want all these single cell tools to be able to use Zarr instead of HDF5, possibly in addition to. We've added support for Zarr to ScanPy, which is one of the popular single cell tools, and that's merged in and released. So it can read and write Zarr files in cloud storage. There's some low-hanging optimizations around how like cloud store seeks are slow, and more work to be done there. So it's not like the main path that people would want to use yet. And then some of the other tools are written in R and haven't totally figured out our story for leveraging some of the same work or over there, or just redoing it in R in some sense. We've also done some work to improve the tooling around HDF5 and try to see if it can be made better before trying to tell everyone to just switch to this other thing, or just ideally have an abstraction where they don't have to worry about which of them that they're using. So Tom, my colleague, took the Java implementation of the parts of HDF5 that conform to the common data model spec and swapped in the Java NIO interface underneath its file system IO. And there are Google Cloud and Amazon S3 adapters or implementations of the Java NIO interface, so we can read and write HDF5 files in these clouds, which to my knowledge is the only place where that capability has existed. And then he also put Spark bindings on top of that, so we can at least read from these HDF5 files that have like chunked arrays inside them. We can just drop workers at specific places inside this giant file that we've split up and make it look like essentially a Hadoop input format or a Spark-friendly parallel read path. So that's cool. It's also like, yeah, not super optimized and not really something we think is going to make that much sense long term, but important technical diligence, I think, to do at the very least. So we want to make all these analyses run in parallel also. Tom created this library called Zappy, which is just like a NumPy array interface that is backed by a variety of interesting distributed processing engines. So Pywren is basically just AWS Lambdas. If you haven't seen it, it's a very interesting project. Like you give it your array and the transformation you want to apply, and it just farms that out to infinity Lambdas and then collects the result back. So I'm really like excited about that as a paradigm going forward. And then more standard like server full distributed engines like Spark and Beam. And you can run it in local mode too. So it's a cool abstraction and library, and it's also released in this Python single cell library, ScanPy. And then here's like some benchmarks running it with Spark that just are kind of 3 different views on the same underlying idea, which is like if you hold the job duration fixed, then you can increase the number of cores and increase the number of cells. Or if you hold the cores fixed, you can get a greater speed up versus a single node version by running on more cells. Or if you hold the cells in the data set fixed, then you can reduce the job duration by adding more cores. And there's some amount of fixed costs to lifting into the distributed paradigm as there always is, but it's about the kinds of trade-offs that we would expect, and we think it's good to give people the option at least to distribute these jobs. Dask is kind of the original version of this trick, which is like it looks like a NumPy array, but we're going to like run it in a distributed way on a big or medium Kubernetes cluster, and we're going to make that very easy. And so we've added support for that to ScanPy as well. So this library is something I've been working on, which is a Zarr implementation in Scala that also wraps this Java HDF5 common data model implementation, so we can convert data sets from HDF5 to Zarr directly in the cloud in parallel. It's because I'm compiling my Scala to JVM bytecode and to JavaScript. I have a web app where I can just read the Zarr data directly like in the browser and have just a completely static thing that could even run some of these conversions if you wanted to, and I don't have to ever see any of your data or anything, or it can proxy the server. And I think that portability is interesting in general. And I kind of went crazy with some like type system and functional programming things in this Scala library, where it's like uses higher kinded types so that you can plug in different multi-dimensional array implementations and parallelization engines. It's type safe in the number of array dimensions, so it knows if you are dealing with a 4-dimensional array or a 3-dimensional array, and if you index 4 dimensions deep in a 3-dimensional array, it will be a compiler error. And it's kind of using the implicit resolution mechanism of the Scala compiler as a kind of formal verification. So it's like a style of programming where you're kind of telling the compiler, here are functions that take certain types to other types, and here's the types I'm starting with, and here's the type I'd like to get to, and like you figure it out. So like program synthesis kind of thing, which is like hard, but maybe good for some things. So anyway, if that's interesting to you, I'm happy to talk about that afterwards. Stuff we'll be doing next. We want to benchmark on bigger data sets. I think there will be public data sets that are larger soon, probably, but we might just synthesize some in the meantime by like just taking the million-cell data set and concatenating it with itself a bunch of times. We want to push further in the processing pipelines some of this parallel and cloud friendliness. To date, we've mostly done like pre-processing and QC steps that are more like map stages than shuffle stages, but there's like clustering and distributed search and indexing things that we'll want to do, especially as there are just like more cells in the world that we want to be able to analyze. And so those algorithms will be interesting, and parallelizing it should be an interesting challenge. The ND-array Scala library I mentioned has just a lot of basic stuff that I need to finish to get to like a V01. One thing I'm very excited about is that in addition to targeting like the JVM and JavaScript, there's a native Scala project, a nascent Scala project called Scala Native that compiles Scala code to native code so that you can interop with C libraries and even have other things interact with those compiled outputs as native code. And then we need to also give some of the same treatment to some of the R-based tools. Probably the easiest way to do that is just inject a Spark R layer into them, but it would be nice to reuse some of these other abstractions we've created that allow for using different parallelization engines and stuff. There is precedent in some of these R tools for like shelling out to a Java jar for some things that aren't implemented in R. There's also precedent for removing that because it was gross. Yeah, it would be cool if I could compile my Scala library to native code that I could call from R. That might be totally insane, but I'll probably try it at some point. Yeah, okay, so summary, trying to make the storage and processing of this single cell data more cloud-friendly and parallel. There's a couple existing tools we want to work with. They span a couple languages, which poses interesting challenges. And there's also multiple good parallelization engines on the market that we want to be able to support. The simple stages we've done so far are scaling about as you'd expect. Some of these things are embarrassingly parallel, and we can embarrassingly parallelize them, and it works pretty well. And then two interesting libraries that we've made as part of this are Zappy, which is this interface for a bunch of parallelization engines over NumPy-like arrays, similar to what Dask is already pretty well known for doing. And then this Scala ND-array library that supports HDF5 and Zarr and has some interesting type safety properties associated with it. So that's all for me. I'll take questions if we have time.
I'm going to use my privileged host position to ask this question. Can you say a little bit more about how much better Zarr is than HDF5? And the second part of that is, do you have an intuition if the Zarr idea or approach is close to what is possible?
Okay, interesting questions. How much better is Zarr than HDF5? Depending what you want to do, it could be infinitely better because it lets us do things that we just can't do with HDF5. I mean, subject to the caveat that you could write an HDF5 library that plays more nicely with the clouds and stuff. I don't know, but it's really complicated to do that. And the path of least resistance so far is to just do something that's simpler, slightly from scratch. An interesting part of the story is that Zarr has now connected with the common data model folks, and they're just kind of like, this looks like it should just be one of our many backends to this unified model of these multidimensional group and data set structures. So probably what will happen is it will just be that it will become NetCDF will be this abstraction over these kinds of meta formats for storing this data or whatever. So, and then your second question was, is it close to optimal for some things? Certainly today, probably not. I mean, I know that we're really focused on kind of this higher level. Let's just we're already in the Spark realm where we're not pinching CPU cycles or being we're not being very precious with our resources. It's more just can we just put more machines at the problem and know that at least we can easily scale, and then we'll optimize it later as we need, which is a choice, not always the right choice by any stretch depending on your context. But I don't know if that's,
Yeah, good enough. We'll talk more about it.
Sounds good.
Yeah, so you covered a lot of relationship with parallelizing arrays, and why not be problem with parallelizing NumPy. It's just over at serializing and deserializing, so even just Dask manage a lot of problems with that. So these tools, how do we get around that? They just want to pull away into Spark and then deal with it, or do we have a difference?
Okay, yeah, so the question's about just overheads that exist with Dask for serializing and deserializing. Those things also exist with Spark for sure, and it's a pain point, and there's lots of people that run benchmarks that are okay, I can just do do this computation on one machine or like 100-node Spark cluster. And so you can definitely get into situations, you have to track those overheads. And I know particularly like PySpark has introduced an extra layer of that historically and had worse performance than like the JVM native Spark bindings. There's a lot of Spark 2.0 was like mitigating that so that the data in the buffers that goes over the wire is like used in place, and they're managing their own memory. And so that's like one way that a lot of these systems get around it. The Arrow project is also relevant for doing things like that. So the short answer is like, we're just piggybacking on what exists in the ecosystem around that. And like, yeah, you have to, if it's a small data set or like you're willing to wait longer, but you really want to minimize the number of CPU cycles you pay for, then like probably don't use all this heavy machinery for it, right? Sure.
Is it too early to ask how much adoption among the scientific community you've got in this project?
No, is it too early to ask how much adoption these things have gotten? Never too early to ask. I think one thing we're trying to really do is just like play nicely with the tools that everyone's already using. And so in the sense that these things are already released in ScanPy, that's like a good, I don't know, at least probably 1/3 of the actual like people in scientific context that are running these tools. Are they using setting the flags to do this stuff today? I would guess not. But I think some of this is forward-looking. We expect the data sizes to grow more. It's still at the stage where, like, if you follow the GitHub repos for these tools, there's like a steady cadence of people coming by and being like, oh, I tried to run it out on this 1 million cell data set on like a machine that had 500 gigabytes of RAM in the cloud, and it still seemed like it ran into some memory pressure. People are still trying to make it work with the data set sizes they have in the single node model. I don't think that they're aware or necessarily that interested right now at incurring the overhead of using these more complicated tools, which is probably correct. But if the data set sizes get 10 times bigger, then they'll probably have to. And so we want to be ready for that. And also, if we optimize this stuff more, it could be like more clearly just better now for the stuff that's on the larger side today, I think. Scott?
Do you think people haven't been generating large data sets because the tooling wasn't there to process them?
Have people not been generating the larger data sets because the tooling was not there to process them? Great question. And that's something, yeah, I've done a lot of like trying to go into these bioinformatic contexts and be like, let's at least have the option to process larger data, because we would probably start asking different questions and doing different science potentially if we did. Which is kind of like, I mean, whatever, it's a self-serving shtick in some ways, but I think it's true. It's hard to characterize this negative space of where we're not looking. I think for sure that's the case. Like some of these analyses that people run today, the first thing they do is just like put some hard cutoffs on the kinds of genes and cells they want to look at to just reduce the data sizes by 10x or 100x to make it just more tenable. And they're probably right. Like those probably was the least interesting 90% of the data, but it's like it wasn't done because it was like good science. It was done partly due to computational constraints. I don't think that all the way at the top of the funnel here, at the sequencing level, they're sequencing less due to this. There's definitely still like there's like an arms race to see who can. Like it's a pride and prestige thing, and it is very impressive to like run these assays at this kind of scale. So to the extent to which anyone is going to be able to do a 10 million cell data set, I'm sure they're racing to do that. And then the tools as they currently exist would probably just start by dropping a bunch of the data and then just working on what's left or something naively.